Hello, everybody. We will start to discuss gene regulation in this chapter. Gene regulation is the mechanism that controls the expression of genes. So, why do we need tight control of gene expression? I want you to think about one basic thing. Take neuronal cell, take muscle cell, take, let's say, epithelial cell, and isolate DNA and sequence it. Would you find differences in DNA sequence? No. Every single cell in our body, of course, with exception of gametes and white blood cells, every other cell has the same genome. However, you see how different the cells are, what a different function do they perform. So obviously there is difference between them, and the difference lies in the gene regulation of gene expression. So if you analyze mRNA and if you look for which genes are expressed, you will find differences. So gene regulation control which gene in which cells at what time is expressed, and it is very tightly controlled mechanisms and systems of mechanisms which controls this gene expression, and we will start to discuss that in this chapter 16, which focuses on gene regulation in bacteria. As mentioned before, the expression of gene in any cell in our body or in bacterial cells is tightly regulated. And the expression, whether the genetic information will be expressed or not, depend on signal which cell receive. And if you look at any particular cell, at given moment, only a subset of genetic information is expressed. So what does it depend on? It depends on signal. It depends on a requirement for survival, for growth, for metabolism. Let's look in bacteria. E. coli is a bacteria which reside in our intestine, large intestine. And basically this bacteria depends on what we eat for nutrition they obtain, they have present. Bacteria can synthesize amino acid, they can synthesize many enzymes, however, and vitamins. However, their synthesis depends on the incoming product, incoming food sources, and therefore the environment, meaning the gut environment, meaning the product which we provide for them through feeding, through our food intake, dictate which gene will be expressed in bacteria. So simplifying it, bacteria respond to environmental changes by altering biochemical pathway through the regulation of their operons. Eukaryotes experience different dilemma. You know that we all start as one cell, as a fertilized egg, which turns into zygote and undergoes the embryonic development, during which millions and millions of cellular division happen. So through the embryogenesis, the genes are turned on and off, and this subsequent functional turning gene on and off regulates the embryonic development. Even if we are mature organism, our cell replicates, and there is differentiation. You recall I use the example of different cells in our body, such as if you compare neuronal and epithelial cells. So each of the cells is specialized. So the specialization requires expression of different set of the genes. So they also, in addition to specialization and differentiation, our cells also respond to environment. And in eukaryote, the levels at which gene expression is regulated are much more pronounced than in bacteria. We have many different levels of gene controls in eukaryote, but we will talk about that later on in chapter 17. So we will now focus on genes, how they are regulated, and what are regulatory elements first. So what is the difference between genes and regulatory elements? First, let's define genes. What is the difference between structural and regulatory genes? This difference relates in their function relevant to gene expression. Structural genes are sequences in DNA which encodes protein which function within cells as a structural protein, as an enzyme, which are part of metabolic and biosynthetic pathways. Regulatory genes are those genes which encodes protein functioning as a regulators of gene expression. So these proteins regulate expression of other genes. In addition, we have regulatory elements. Regulatory elements affects the expression of other genes. They are sequences in DNA which are not translated but still regulate expression of other genes. They can either enhance or inhibit expression of other genes via the interaction with protein which are involved in enhancement or suppression of gene expression. Now, how the genes are regulated? Before we go there, I want to point on one thing. In previous slide, I mentioned that gene expression is regulated by signals received by cells. Now, keep in mind that this does not apply to all genes. We have group of genes which are constitutively expressed. These genes do not undergo gene expression regulation. What belong to this group of constitutively expressed genes? They are so-called housekeeping genes, which are genes encoding proteins important for maintenance of the cells, for cell survival, for normal cellular function, such as gene which encodes actin protein, which is cytoskeletal, or gene which encodes tubulin proteins, ubiquitin protein, and so on. So those genes are constitutively constantly expressed. However, most of the genes are regulated. The expression of most genes is regulated, and they might be under either positive or negative control. What does it mean, positive control? It means that this proteins stimulate gene expression, or positive control stimulate gene expression. Negative control inhibit gene expression. If you compare gene expression in prokaryote and eukaryote, you will find many similarities but also many differences. And if you look at the control of gene expression particularly, you will see that in prokaryote most of the genes are negatively controlled, while in prokaryote most of the genes are positively controlled. Regulation of gene expression in bacteria and eukaryotes is predominantly accomplished by proteins that bind to DNA sequences and activate DNA or inhibit DNA, so-called DNA regulatory proteins. We call them DNA binding proteins. What is common for all these regulatory proteins are the discrete functional parts which are called domain. Some of the motifs within domain, binding domains, are outlined in this picture. So what is characteristic for this domain? They have motifs. You can see here 3 different motifs. You can find these motifs in domains in bacteria and eukaryotes, such as helix turn helix motif, and there are motifs within this domain which are particularly and specific for eukaryotic transcription factors, such as leucine zipper motif, and in many eukaryotic proteins you can find also zinc finger motifs. So what are this domain? What is their function? The domain of DNA binding regulatory proteins is to interact with DNA and either turn off or turn off gene expression, as I mentioned before. So where do they bind? Most of the DNA binding proteins fit into major groove and bind, either forms a hydrogen bonding with the bases, or they might also interact with the sugar phosphate backbone of DNA. But as I mentioned, majority of this regulatory protein bind with the major groove. Now, how this binding is accomplished? It is dynamic. It is on and off. Transcription factor, let's say one example in eukaryote, transcription factor binds to the promoters when gene has to be expressed. However, you do not want to have this transcription factor bound permanently because you would lose control of the expression. So, as I mentioned before, the binding is either hydrogen bonding or other non-covalent interaction. So the binding of transcription factor and other DNA binding regulatory proteins is dynamic and always non-covalent. So let's now discuss control of gene expression, control of genes transcription in bacterial cells. As I mentioned before, there are many similarities, but there are also many differences in the gene expression control in bacteria and eukaryote. One of the major differences is that in eukaryotes, each gene has own promoter and it's transcribed separately, while in bacteria, control of gene expression lies in the organization of functionally related genes into operons. Within operons, several related genes encoded proteins which are related or which function in one metabolic pathway are transcribed from common promoter and form so-called polycistronic mRNA. So, in bacteria, single promoter transcribes several genes into a single mRNA, which is polycistronic. Now, so what is operon? Operon consists of, let's try to outline it here. Operon consists of the, of course, promoter. Afterwards, we have the structural genes, let's say gene A, gene B, gene C, and between at the end overlapping promoter and structural gene is so-called operator. So this is operon. Okay, now where do the regulator genes come? Because operons are controlled by regulatory proteins. So usually upstream of the operator, so somewhere here, we have regulatory sequences. And these regulatory sequences have own promoter and they encode regulatory protein. So this regulatory protein which is controlled from the own promoter act on operator. And we will now discuss what kind of operon we have and what kind of regulation we do have. So let's now talk a little bit more in detail about operons. You can see here you have operon structure right here, and here you have regulatory protein. So regulatory protein is synthesized from regulator and it is under control of the own promoter. Now, what can be regulatory protein right here? It can be either a repressor if the control of operon is negative, or it can be an activator if the control of operon is positive. So negative control means that regulatory protein repressor binds to operator and suppresses operon function, gene expression. Positive, an activator, binding of activator to operator would trigger or activate the transcription gene expression from this operon. So negative repression, positive activation. Now, this is regulatory sequences, regulatory protein which can regulate operon positively or negatively. Now, operon can be either inducible or repressible. Inducible operon, what does it mean? Just think about it. If something can be induced, it's inducible, so it means that it's normally off, but it can be turned on. Repressible operon, contrary to it, if you can repress something, it means that it's normally on and you can turn it off. Now, whether the operon is inducible or repressible is depending on how the regulatory protein is synthesized, whether it is in active or inactive stage. And we will come to it a little bit later. What I would like to point at this point is that inducible operons function usually to control expression of genes involved in the synthesis of proteins involved in breakdown pathways, metabolic pathways, such as lac operon. Lac operon in bacteria control breakdown lactose into glucose and galactose. So it makes sense for bacteria to not waste energy keeping inducible operon on, such as lac operon on, if there is not lactose present. So it is more economical just turn this operon on when there is a substrate which needs to be break down. Contrary to it, repressible operons are usually on, and they are on because there's a proteins synthesized by these operons are involved in the synthetic pathways, such as synthesis of amino acid, let's say TRP operon. Since bacteria needs synthesis of certain amount of amino acid continuously, it's much more economical for bacteria to keep the synthetic operon such as TRP on, and only if there is enough accumulations of the amino acid, let's say, or other final product to turn it off. In most cases, repressible operon functions through feedback mechanism where terminal product of the operons is actually regulates the repression of the operon. So let's now talk a little bit in more detail and characterize what is a negative inducible operon, negative repressible, and positive inducible and repressible. We already know what is the difference between inducible and repressible operon. So you know that inducible operon is normally off and it needs to be turned on, while repressible operons are normally on and they needs to be turned off. When you want to characterize an operon, you always break it down to two parts, whether it is positive or negative regulation and whether it is inducible or repressible. So we talk about inducible, repressible. Now, negative or positive. Negative regulation means that regulatory protein is repressor. When repressor bind to operator, it shuts down the operon. In positive control, the regulator protein is an activator, and binding of an activator to operator is required for operon expression or for transcription of gene in the operon. So you know that in bacteria predominantly regulation is at the level of negative inducible or repressible regulation. So let's talk first about those. Let's talk first about negative inducible operons. So it is negative. It means that regulatory protein is repressor and it's inducible. So you know that it's normally off, but it can be induced on. So what does it tell you? Repressor proteins are synthesized constitutively. So this repressor, since they can keep the operons off, it means they have to be synthesized as active molecule, active repressor, which can bind to operator, and this binding would shut down the operon. So, negative inducible repressor is synthesized as an active molecule which can actively bind to the operator and keep operon off. So how it can be induced? You will need small molecules, so-called inducer. Inducer would interact with repressor, change conformationally repressor. So now this conformational change would induce such a change in shape that the repressor cannot bind to operator. So now once the inducer is actually changing repressor cannot anymore bind to operator, so the operon can be turned on. What about negative repressible? Let's step back a little bit here. So if you have negative repressible, what does it mean? Negative, it's still repressor, but it's repressible, so it's on. What does it tell you? The repressor which is synthesized is inactive. It cannot bind to operator and shut down the operon, so operon is on. However, it can be repressed. It can be repressed if there is enough of small molecules which are called so-called co-repressors. This co-repressor works similarly as inducer in a way that they bind to repressor and change its conformation. However, difference is that co-repressor would activate repressor. Upon interaction with co-repressor, repressor now can bind to operator and shut down the operon. So, negative inducible, it means repressor is synthesized in active form where it can bind to operator and shut down operon, and it needs to be changed by inducer so the operon can be induced. In negative repressible, repressor is synthesized in inactive form, so it cannot bind to operator. It needs co-repressor to change its conformation to that which can bind to operator. Positive regulation is very similar. Only difference is that regulatory protein is an activator. It means upon binding of an activator to operator, the gene expression from operon is on. Otherwise, it's very similar to negative inducible, negative repressible control. What would I mean by saying that it's similar to negative inducible and repressible? I refer to inducible and repressible parts. So if you have positive inducible, it means that in this case, activator is synthesized in inactive form because it cannot bind to operator and activate transcription. So, but it can be induced. So the activator can interact with small molecules which can change its conformation, and now it can bind to operator and activate transcription. Positive repressible, it means that activator is synthesized in active form, so it binds to operator and turns transcription on, and it can be repressed if activator interacts with small molecule. So here is an example, the lac operon of E. coli, an example of a negative inducible operon. So you see that regulatory protein is synthesized as an active protein which binds to operator and prevents RNA polymerase from binding and transcribing the structural genes. If you have inducer present, in the case of lac operon is allolactose, this inducer changes the conformation of the repressor, so now it cannot bind to operator, so RNA polymerase now can bind and starts a transcription. Here we have an example of negative repressible operons such as TRP operon in E. coli. In negative repressible operon, the repressor is synthesized in inactive form, so it has such a conformation that it cannot bind to operator. This enables RNA polymerase to bind to promoter and initiate transcription of structural genes. So the expression of these genes is on until there is accumulation of the final product. Once there is saturation of final product, this final product can function as co-repressor. It can bind to repressor, change its conformation. So now repressor can bind to operator and prevents RNA polymerase from binding to promoter and initiating transcription of the structural genes. Remember that many negative repressible operon are regulated by feedback mechanism when the final product, accumulation of final product can trigger a silencing of repression of the operon. So here is just summary of what we have been talking about. Positive and negative control. Here is negative, positive control, inducible and repressible. So what I want you to keep in mind is positive control means the regulatory protein is an activator. In negative, regulatory protein is a repressor. Inducible, the operon is off, but it can be induced on. Repressible, the operon is on and it can be turned off. And this inducible and repressible apply to both, whether it is positive or negative control. The difference between positive and negative is in the regulatory protein, whether it is activator or repressor. The lac operon of E. coli was the first operon described and defined by Jacob and Monod in 1961. They studied lactose metabolism in E. coli and prepared series of mutants and studied their interference with normal lactose metabolisms. Based on the studies and data they obtained, they were able to propose a structure of lac operon. And the structure is outlined right here. The operon consists of lac P, which is promoter, lac O, which is operator, lac Z, which encodes beta-galactosidase, lac Y, which encodes permease, and lac A, which encodes transacetylase. So this operon is controlled by regulator repressor, which is encoded by lac I. Now, lactose. We know that lactose is major carbohydrate in milk. When we drink milk, our E. coli in our guts can actually break down this lactose into galactose and glucose. And you know, glucose is major source for the ATP production. Now, how all this happen? Firstly, lactose has to be actively transported into bacterial cells, and this active transport is mediated through permease, which is encoded by lac Y. It's permease. Once the lactose is inside of cells, beta-galactosidase encoded by lac Z, so this is beta-gal. Beta-galactosidase breaks it into galactose and glucose. In addition, it also synthesizes, besides galactose and glucose, it also synthesizes allolactose. And you know that allolactose is an inducer, which induces lac operon. Function of lac A, which encodes the transacetylase, is not clearly identified. So, what happens is when we drink milk, as I mentioned before, there is excess of lactose in our gut, so bacteria brings lactose in cells, via permease activity and convert it to glucose and allolactose, which upregulates lac operon. Now, there is one problem. If lac operon is repressed, if it's not working, where is the permease? How lactose get into the bacterial cells? So the answer is very simple. The repression of the lac operon is never complete. It's never completely shuts down. So there is always background transcription which generate enough permease that when lactose is present, it can be transported into cell. Of course, once that happen, synthesis, lac operon activity increases and synthesize more permease, more beta-galactosidase, and basically the activity is enhanced. So let's now summarize how the lac operon works. We know that lac operon is negative inducible. So the regulatory protein is a repressor which is synthesized as active protein which binds to operator and prevents transcription of the operon. So there is no transcription, respectively, there is no efficient transcription, there is only very low basal transcription. What happens when the lactose is present? You know that basal transcription enables very low levels of beta-galactosidase and permease being synthesized, which actually bring that lactose into cells, and beta-cal actually processes break it down to glucose and galactose, and at the same time it also synthesizes allolactose. Allolactose functions as an inducer. It would interact with a repressor, change its conformation, so now repressor cannot bind to the operator, so RNA polymerase now can efficiently bind and trigger transcription of genes, so the activity of the operon is significantly enhanced.